the introduction. To make the introduction shorter, you can call me a bacterial psychologist. After the first speaker, who took more the approach of a medical person interested in patients, I am much more interested in the bacteria and understand who they are and when and why they're doing well. What I want to do today briefly, I want to briefly go over as the first speaker on who are the organisms in our gut, what are they doing. In terms of the probiotic applications, I will focus more on their use in food than on the medical use, including the question which health claims can you actually put on foods or supplements containing probiotics. Some factoids on intestinal microbiota. We are more bacterial than humans. Our body has 10 times more bacterial cells than human cells. If you do the counting on the number of genes, it's 100-fold. And most, not all, but most of these bacteria live in the digestive tract. And in the last couple of years, we have started to appreciate gut microbiota connectivity as an important organ, which has metabolic and immunological functions. And in its function, you can compare it to heart, liver, lung, and other organs which are known to be quite important. Where are they? They are mainly in the colon. In the upper intestinal tract, we have secretions of bile acid, which are bacteriostatic or bactericidal, and there is a swift transport of nutrients. So relatively few bacteria. Here in the colon, where the digesta stay for some time, we have high cell counts, 10 to the 11 bacteria per gram. 10 to the 11, that's 100 billion bacterial cells per gram. So quite a large number, and feces actually is half of that is bacterial biomass. One problem with the organisms in our gut, we can't do to them what we can do to many other organisms we're interested in. We cannot grow them in pure culture in the laboratory, and that made it difficult to investigate their properties until probably 10 years ago, when DNA sequence-based technologies started to become widely available and suitable to study which organisms are there and what are they doing. Without putting too much science into the talk, I want to explain some of the names that you will come across quite often if you read about the bacteria in the intestine. You see here bars, and the bars are drawn to scale, which means a large bar means they have a big contribution to the organisms, and a very small bar means they're essentially insignificant. And if you know three names, you can pretty much account for 99% of all the organisms, and that's Firmicutes, which is a relatively a broad term for organisms which are gram-positive. Most of these are strict anaerobes. Bacteroidetes, also quite abundant, that's gram-negative organisms. Both groups are specialists in fermentation of polysaccharides. And then we have Bifidobacterium, which is frequently used in probiotic preparations, and pretty much all the rest, including Lactobacillus, in terms of numbers, is quite insignificant. Why are they so important for us? We do three things which are quite crucial. The first is they feed us. We can digest sugar and starch, and pretty much any other carbohydrate in the diet is indigestible for us, which means in the absence of intestinal microbiota, we would lose 10 to 30% of our metabolic energy. If you take a Canadian perspective, 10% less may be a good thing. Go 100 years back and 10% more or less made a difference in terms of being able to survive. So this is actually quite an important point. And then the intestine is a surface which separates outside from inside. And our intestine is exposed to more foreign material than our skin, simply because we eat all the time. So it's also a big site where our immune system monitors and watches out for invading pathogens. And the commensal bacteria, the non-pathogenic bacteria, are very important in triggering the development of the immune system and many autoimmune diseases, allergies, inflammatory bowel disease, diabetes are linked to lack of exposure of the immune system to parasites and commensal bacteria. And then they protect the host by competition with pathogens, and together who they are and what they do can be quite decisive in health and disease. Dr. Kroeker indicated that they can be quite stable over time. It's also that each of us has his or her own characteristic composition of organisms. Each person carries about 400 species, but which organisms are there is different from person to person. And one of my colleagues, I cannot call that a footprint, because the fingerprint representing the bacterial community in the gut is as characteristic and as stable as a fingerprint of a person. So you could replace the fingerprint by something which is here, which is essentially a fingerprint generated by PCR, a fingerprint of the bacterial organisms in our gut. What you see here is a pattern, each band representing one bacterial species. The more similar the patterns are, the more similar the bacteria are in the gut. And this was a study which used homozygotic twins. And you see that if you compare two twins, they have the same genome, the patterns of the gut bacteria are very similar, not identical, but very similar. That tells you that our genome has a decisive influence on who they are and what they do. If you compare people from the same family that may have a large overlap in lifestyle and diet, you see that the patterns are very different. So diet, lifestyle also influence, but to a lesser extent when we compare to the host genome. Why is that so? One of the biggest determinants of the gut microbiota is the host immune system, and that goes both ways. Gut bacteria influence very strongly the host immune system, but also the host immune system secretes molecules which change the composition of the organisms in the gut. On top of that interplay, we have, of course, the food that comes in, which contains bacteria and non-digestible carbohydrates, which are substrate food for our bacteria, and we have competition and interaction between organisms, and all this makes a quite complex interplay which is not easy to understand. A definition of probiotics, Dr. Kroeker had that on her slide again. We call a preparation prebiotic if it is viable bacteria in high numbers that change the microbiota in the compartment of the host, and they must have some beneficial property. Organisms which are frequently used include Lactobacillus and Bifidobacterium. I tried to convince you previously that Lactobacillus really isn't an important part of human gut microbiota. It's different for some animals, but then we are not animals. Why are they used as probiotics? One reason is that Lactobacillus has been used in food production since the onset of agriculture, around about 10,000 years, which means humankind has been eating Lactobacillus by the billions per day since 10,000 years, and they hardly made ever anyone ill. So if an organism is safe, that's about as safe, as sure as we can be that an organism is non-pathogenic. Because it's so important for food production, we also know the organism very well. If there is economic importance, there is some incidents for scientists like me to study them. So we know them very well, which means we can influence them, grow them, and predict their behavior in a number of situations. It's short. Second connection between Lactobacillus and probiotics dates to the origin of the concept. The concept of probiotics isn't terribly new. It goes back to the beginning of the 20th centuries, which was pretty much when microbiology was still an emerging discipline with exciting discoveries every other year. And in this time, a Russian scientist working in France suggested that we can help and improve digestion by eating beneficial bacteria. And that observation was based on anecdotal evidence that Bulgarians living in the Caucasus, they live very long and they also eat a lot of yogurt, so probably there must be a connection which could be linked to the consumption of milk fermented with lactic acid. That connection pretty much stuck. We still associate probiotics frequently with yogurt. We need to keep in mind that probiotic is not only a clinical aspect related to treatment, it's also big business in the food industry. The food industry has been using it in the last 20 years pretty much to tackle the price of yogurt. If you buy a plain yogurt which is just fermented and tastes well, you pay much less than if you buy a yogurt which contains probiotic bacteria. The probiotic segment still has strong growth rates. If you look at China, Brazil, USA, the market is growing quite strongly. It's no longer growing in Europe. There, the probiotic products were introduced first in the 1990s, and the growth has taken place before 20. Why are probiotics working? I tried to convince you previously that the biggest determinant of which organisms are in the gut is the host immune system, and all evidence that we have now indicates that the biggest contributor for health benefits of probiotics is their stimulation of the immune system, which means probiotics are foreign to the host. They're recognized by the immune system, so the immune system has to do something. They're recognized as something which is not a pathogen, but still this response of the immune system to the incoming bacteria leads to an improved alertness of the immune system, which helps fighting off other diseases. So the immunomodulatory activity of probiotics is probably the single most important determinant of their beneficial activity. What probiotics don't do, they are not stable, they will not become a stable part of the organisms in our gut. This has been shown nicely in the early days of probiotic work in around about 2000. What that study did, that was confirmed by a number of others. They looked at lactobacilli in people before they consumed the probiotic preparation, during the consumption of probiotic preparation, and afterwards. And the strain called BR20, which is black, is the probiotic. And what you see, of course, before they consume the probiotic strain is not there. While the probiotic strain is consumed, yes, you find it, not in very high numbers, but you find it. But as soon as you stop eating the probiotic organism, you no longer find it in the gut. The organisms that have been there before are likely to take over again. So it's always, they're transient, they never colonize permanently. If you want to apply probiotics in either food or supplements, you always have a problem, and the problem is your organisms, you need to get them to the patient. The gut is quite different from the outside world. There are essentially two strategies. Either you put the probiotic organisms into a food. In most cases, companies use fermented food, which means you take the lactobacillus or the bifidobacterium in yogurt, and you hope that it survives. Yogurt contains a large amount of bacteria, but they may or may not have probiotic activity. And here your biggest problem is get the probiotic bacteria to survive in the food system while the yogurt is sitting on the shelf in the supermarket. Keep in mind, by definition, probiotics are live bacteria. So if they die before you eat the product, they may or may not work anymore. A second possibility is you pack the probiotic organisms in a pill, which makes it a little bit easier to keep them alive. It's still quite a challenge, but now you need to talk your consumers to eat pills, which is not as nice as forcing them into eating yogurt. So survival and activity of probiotic strains during production and storage is a major challenge in the field. And the most successful strains are those where the companies have been able to solve this problem most convincingly. What are the health benefits? The list is increasing. The well-documented health benefits, where you have a large number of studies which show the same results, is shortening of rotavirus diarrhea, less travelers diarrhea, and alleviation of lactose tolerance. There is a much larger group of things under investigation. Mind, as soon as it says treatment, you benchmark against the use of drugs, which means your level of scrutiny to demonstrate an effect is very different. If you're treating disease, you need to show activity comparable to a drug, which is a very high level of scrutiny. Possibly the data that we have for most of the diseases is not yet good enough to replace other chemicals as a drug. If you use it as a food supplement, you don't treat disease, you sort of try to sell a food system to a consumer, which is a very different story. Mind that in most cases, if you're in the business of selling yogurt or fermented drinks or whatever, you don't try to sell to people that have IBD, or you don't try to sell to people that travel to Mexico, but you try to sell it to everyone. So in most cases, you have very general statements. They normalize the balance of the flora, intestinal cleansing, found in one yogurt, stimulate the immune system, whatever that means, and they enhance the immune system response and keep the intestinal flora in good shape. So very, very general statements, somehow associated with a better well-being. And since a few years, Health Canada started to regulate the claims on food. So these are statements that you can actually make in Canada. Naturally forms part of the gut flora. There are live organisms that naturally form part of the gut flora, contribute to a healthy gut flora, and so on. So what you find in the food applications or in the supplements is no disease claims. They don't exist. They haven't been shown at the necessary level of scrutiny. But very general statements, they somehow make you feel better, and that has something to do with the bacteria. If you apply probiotics, and there is not a whole lot of regulation, there is a lot of scientific consensus, and that goes back to the definition. You need to have live microorganisms in sufficient numbers, and you need to have a health benefit, which means to get a probiotic preparation well documented, you need somebody from medical sciences who is able to document the health benefits to humans. That's somebody like Dr. Crocker, who works with patients and can say whether or not they feel better. But you also need a well-characterized bacterial strain who is known, very well characterized and viable, and that's then sort of my expertise. Who are they and what are the bacteria? I will skip a little bit to keep the time. One question, of course, companies sell defined strains, and they did the clinical trials to show health benefits. But if you look at the supermarket shelves, there is a whole lot of foods which contain a large number of of lactobacilli. And if you want to buy Lactobacillus acidophilus, buy long-ripened cheese. Lactobacillus thorntonii, you will find in kefir. You can buy that cheap in the supermarket. Lactobacillus reuteri, the pure strain, you will find often as a probiotic. If you want to buy it in the supermarket, you can also buy cheese, a number of sorghum products, most of them not available here as well. And the list goes on, which means for most of the lactobacilli which are used as probiotic cultures, they're also used in one way or another in food production. The question is, do these food, as they're produced, as they have been produced in a couple of thousand years, do they have beneficial health effects on our health? Probably yes. Has it been shown? No. Simply because the margins don't justify the expense for the clinical studies. But probably many of the natural fermented foods have comparable health benefits as some of the defined that brings me to the summary. First, going to the intestinal microbiota. They're stable and they're important for our health. And at some level, their composition is something which is very individual and different from each person. We can not alter for long-term the composition of the intestinal microbiota, but by eating live cells of probiotic bacteria, we can alter the function, particularly through interaction with the immune system, and we can eat food which naturally contains probiotics, or we can specifically add some strains to get health benefits derived from our food. That brings me to the end, and I hope we have enough time for some questions. Thank you. Yeah.
Can you say that the immune system attacks the probiotics that we eat?
Attack is the word, is the wrong word. We recognize. We
So
recognize.
it recognizes it, but it doesn't actually destroy it.
It doesn't. But you see, if you eat a probiotic, the initial response of the body is the same response as you would see for a pathogen. Something's new, something's foreign, we have to do something, we have to watch out. And then after some time, there is a recognition, okay, they are foreign and new, but there are pathogens, so we can down-regulate inflammation. But the initial response is the same. You have a certain level of a pro-inflammatory response, which is then dampened because they are recognized as non-pathogens.
So they're not recognized as pathogens.
Yeah.
Oh, okay, because I thought that if they were attacked, they would understand like how they would stimulate the growth of more bacteria.
They don't stimulate the growth of other bacteria. So pretty much that's here. The total of your gut bacteria is 100 billion, and you eat a few million, which means the numbers of probiotic organisms make no difference to the numbers which are there. You have difficulties finding them. You have to use very specific methods. So the bulk of your bacteria in the gut doesn't really change. What changes is, our immune system is well versed to recognize something which comes in. So even if it's a small number. Yeah.
So you're saying that the gut recognizes the bacteria as not as a host. So would you just be extreme?
Well, they go out the way they go into their travel through. Yeah.
Okay.
The slide that showed the permissible statements in Canada that they can make, the middle one talked about them being natural, that naturally form part of the gut flora. But you had pointed out earlier that these are just transient and not there normally. And I'm wondering if there's a misunderstanding.
No, you don't. It depends on which level you look at. For example, bifidobacteria make up to 10% of the bacteria in the human gut. But the specific strain that is used as a probiotic will never outcompete the organisms which are there. So it depends if you look at what we call genus levels, which is a broad, it's like a clan, a large family. Then they're the same. If you look at the strain level, which is looking at one person, then they're different. So at different levels, but the strains, on the strain level, the organisms that come in will never make it as a stable colonization. Although you may find other organisms from the same family or the same clan. Yeah.
Thank you.
One more question? We got you.
Thank you.
Little. Yeah.
Yes.
Thank you.
Thank you for